Here we're going to talk about how cells make ATP. We've mentioned before that ATP is a really important molecule because it's one of the major molecules that cells can actually use directly in order to power its different activities. And the reason why ATP is such a high energy molecule is because it has three phosphate groups. Remember that each one of these phosphate groups has a negative charge to it, so that means that if you have a grouping of phosphates like you have here, they're going to tend to want to repel one another. And so because they have this urge to move away from one another, that's what makes this portion of an ATP molecule have a lot of potential energy. And so you know that energy just doesn't appear out of nowhere. So whenever we say that cells have the ability to make ATP, what we really mean to say is that cells have the ability to take in certain molecules that have high potential energy on their own, and then use those molecules in order to make something else that has a lot of potential energy, for example ATP. So examples of molecules that certain cells can take in and use to make ATP include glucose molecules, or even more generally sugar molecules, and also lipids. And so again, these are two examples of high energy molecules that some cells can use in order to make ATP. What makes things like sugars and lipids have high potential energy is the fact that they have a lot of carbon and hydrogen bonds in them. And so if you can't see the carbon-hydrogen bonds in this glucose molecule, if we redraw it in its linear form, it'll make it easier to see. And again, the reason why carbon-hydrogen bonds are considered to be high-energy bonds is because carbons and hydrogens have roughly the same electronegativity. So that means that if they were to share an electron during a covalent bond, that electron would spend as much of its time around the carbon as it does around the hydrogen. This is considered to be a high potential energy bond because basically what you have here is that you have two atoms, carbon and hydrogen, that's pulling on that electron with equal strength. And so the analogy that remember that we used before is like it's a tug of war. So if you have a tug of war going on between two teams on either end of the rope, and they're both pulling on that rope with equal strength, that means that there's a lot of tension, there's a lot of strain that's built up then in that rope. That rope then has a lot of potential energy, and you would be able to see the proof of that potential energy if you cut it. So if you cut that rope while both of those teams are tugging on it, both of those teams would end up crashing towards their respective ends. So kind of the same idea applies here. Because that carbon and that hydrogen are pulling on that electron with equal strength, there's a lot of tension, there's a lot of strain, there's a lot of potential energy that's stored up in that electron in this particular situation. So that's why carbon-hydrogen bonds are considered to be high energy bonds. On the other hand, if you have an oxygen to hydrogen bond or an oxygen to a carbon bond, those are considered to have less potential energy. And the reason why that is because of the relatively high electronegativity of that oxygen compared to both hydrogen and carbon. So because of that different level of electronegativity, that means that if these two atoms were to share an electron, that electron would be spending more of its time around where the oxygen is. And so because that hydrogen atom has a relatively weak pull for that electron as compared to the oxygen, that means that there really isn't a lot of strain that's being placed on that electron. In other words, there's not a lot of potential energy that's in this bond. So oxygen and hydrogen, whenever they form a covalent bond, it's considered to be a lower energy bond compared to carbon and hydrogen. Same thing with bonds between oxygen and carbon. Those are considered to be low energy bonds as compared to the energy that you find in the bond between carbon and hydrogen. So again, ATP is considered a high energy molecule because it has three phosphate groups on there. Each one of those phosphate groups has a negative charge. Electrons have negative charges. Glucose and lipids are considered high energy molecules because those carbon-hydrogen bonds have high energy electrons in them. So you're seeing here that electrons are kind of important, and in fact if you were to boil cell energetics down to its essence, basically all it involves are electrons. And actually whenever you have this happening, so whenever a cell takes something like glucose and uses it to make something like ATP, basically what's happening there is that electrons are moving, and it's that movement of high energy electrons that allows for the conversion of something like glucose to something like ATP. So here's what we mean by that. Again, we said that glucose is something that can be used by some cells in order to make ATP. What we're saying now is that what causes that to happen is when the high energy electrons in that glucose molecule end up moving towards another molecule that serves as an electron acceptor. And so an example of a molecule that our cells commonly use as an electron acceptor is oxygen. And oxygen is a really good electron acceptor, again, because of its high electronegativity. Right? It's really good at pulling electrons towards it. And so that's what happens when ATP is made. So something like oxygen ends up pulling high energy electrons, for example, from glucose towards it. And whenever anything moves, like electrons, that represents kinetic energy. And it turns out that cells can use the kinetic energy from that moving electron in order to make something else that has a lot of energy, like ATP. So again, it's whenever you're talking about ATP being made, whenever you're talking about cell energetics, it's all about what's going on with the electrons. So to sum up this little slide here, again, the kinetic energy of moving electrons is used by cells to make ATP. So in order to understand then how cells make ATP, it'll help to know a little bit something about moving electrons. And it turns out that there's a term used to describe chemical reactions that involve just that involve the movement of electrons. And so the type of chemical reaction then that involves the movement of electrons, those are called oxidation-reduction reactions, or redox reactions for short. And again, basically what occurs during a redox reaction, it involves, redox reactions involve two molecules, one molecule giving up an electron and another molecule accepting an electron. So that's all a redox reaction is. Again, it's a transfer of electrons from one thing to another. There are terms used to describe what happens to the there are terms used to describe the molecule that gives up the electron and the molecule that accepts the electron. So it turns out that the molecule that gives up the electron is said to have undergone oxidation, whereas the molecule that accepts the electron, that's the molecule that's said to have undergone reduction. And so another way of thinking about this, so because reduced molecules have accepted electrons, that means that reduced molecules tend to have a negative charge. On the other hand, if you're considering the situation with an oxidized molecule, because by definition oxidized molecules have already given up an electron, that means that they tend to have more positive charges on them. And so there are different mnemonics that people use in order to remember the difference between oxidation and reduction. The one that I use is this. The thing that donates electrons becomes oxidized, and so donate has an O in it. Oxidized has an O in it, so that's kind of how I remember what oxidized means. Another little clever mnemonic that some people use is this. Leo the lion goes grr. And so that stands for, Leo stands for if it loses an electron, that means that it's oxidized. Grr stands for this. If it gains an electron, that means that it's reduced. So again, there's a couple of little tricks that some people can use to remember the difference between oxidation and reduction. One thing to point out is that whenever electrons move within cells, it's pretty rare that it's just a naked electron that ends up moving from one place to another. Instead, that electron is usually packaged with something else, and it's that entire package that moves between things inside of cells. And so here's what we mean. Right, so again, in cells, it's rare that it's a naked electron that ends up moving. Instead, it's packaged together with a proton. And so if you remember your basic chemistry, whenever you have a single electron packaged together with a single proton, that's what a hydrogen atom is. Right? So by definition, a hydrogen atom is a thing that has a proton and then an electron spinning around it. So again, in cells, it's more common that a hydrogen atom is the thing that's transferred between molecules instead of just a naked electron. And so you'll see this later on in this video, you'll see this in class, and you'll see this in your textbook, that hydrogen atoms are being transferred between molecules whenever ATP is being made. And again, the significance of that is that the hydrogen atom contains an electron in it. And again, it's the movement of the electron that's actually used in order to make an ATP molecule. It turns out that making ATPs also involves hydrogen protons. And so what we'll mention here is that, remember that there is actually a difference between, first of all, let's sum this up. So in cells, electrons are often transferred between molecules in the form of a hydrogen atom instead of a naked electron. Again, hydrogen atoms and hydrogen protons are both they both play key roles in making ATP inside of a cell. We'll see exactly what protons and these atoms do later on, but for now, we'll ask you to keep in mind that there is a difference between this and this. Right? So they might look the same. They're both H, except that just one has a plus on it, but it's actually a pretty major difference. So they look the same, but they're not actually the same. So remember that whenever you see just an H, that means a hydrogen atom. And again, hydrogen atoms have an electron and a proton in it. Whenever you see this H+, what you're talking about is a hydrogen proton. And so again, these two things are kind of completely different. So one has an electron in it, the other one doesn't. And so because they're completely different, we'll kind of be picky when we're asking you to explain ATP generation in terms of what the difference is between a hydrogen atom and a hydrogen proton. All right, so again, oxidation reduction reactions, that's basically it's a reaction that involves a transfer of electrons from one thing to another. We're talking about oxidation reduction reactions here because it's the movement of electrons that's actually responsible for making ATP inside of a cell. One more thing we'll mention about oxidation reduction is this. It turns out that you can use these terms, oxidation and reduction, to describe molecules either giving up or accepting electrons. You can also use that term oxidation and reduction to describe single atoms that are either giving up or accepting electrons. So here's what we mean by that. So for example, if you consider a carbon dioxide molecule, it's possible to describe this one carbon atom within the CO2 molecule as either being oxidized or reduced. And so here's why you can use those terms to describe this carbon atom. What's holding this carbon together with these two oxygens is the fact that they're sharing electrons. Remember that oxygen has a stronger pull for the electron than the carbon atom does. So that means that this electron that's being shared between this oxygen and carbon would be spending most of its time around where the oxygen is. Same thing here. This oxygen has a stronger pull, so the electron would spend more of its time around where it is. Remember, we said that one way of thinking about oxidized things is that they're things that tend to have a positive charge. And so check out this carbon atom that we're pointing to here. All right, the electrons that it's sharing is spending more of its time around the oxygen. In other words, the electrons are spending more of their time away from where that carbon is. That means that this carbon kind of has a partial positive charge to it. Because that's the case, one can describe this carbon atom in a CO2 molecule to be oxidized. Right? So when we say that individual atoms are also can also be described as oxidized or reduced, that's what we mean. If you contrast that to this carbon atom in this glucose molecule, its oxidation state is a little bit different. And so the reason why that is because of who it's sharing electrons with. So it's sharing an electron with a hydrogen, it's sharing electrons with carbons, and it's also sharing an electron with oxygen. You know that carbon and carbon would be tugging on that electron with equal strength. Hydrogen and carbon would be tugging on that electron with equal strength. Same thing here, equal strength between the two carbons. The only difference of electronegativity would occur between this carbon and the oxygen. So whenever you're considering this particular carbon, it would be the only one that you see in this picture that would be shared unequally. So it would be spending more of its time around where the oxygen is. So now if you compare this carbon atom in the glucose molecule to this carbon atom in the CO2 molecule, you can see why this carbon atom would be considered the more reduced of the two. So remember before that one of the ways of describing a reduced thing is that it tends to have more of a negative charge. So because this carbon atom has electrons spending more time around it as compared to this carbon atom, the carbon atom in the glucose is considered to be more reduced as compared to the carbon atom in CO2. And so you might see in the textbook, and we might mention in class sometimes, that glucose, we might say that glucose is an oxidized molecule. And so whenever you hear that, it might be actually more precise to say that a carbon molecule contains, it might be more precise to say that a glucose molecule contains carbon atoms that are reduced, right? And so again, what makes this carbon atom reduced is the fact that it's bonded to things like hydrogen and carbons with which it's sharing electrons equally. We've mentioned before that carbon-hydrogen bonds tend to be high-energy bonds. So there's a relationship between reduced molecules and high-energy molecules. So molecules that tend to be reduced also tend to have high energy to them. So again, to recap, making ATP inside of a cell, it's all about electrons moving. It turns out that there's some electrons that are involved in higher energy bonds as compared to other electrons. So now that we've kind of gotten that out of the way, we can actually start talking about how cells make ATP. What we'll do is talk about two major ways that cells can make ATP. One way is called respiration, and the other way is called fermentation. What both of these have in common is the fact that basically it involves electrons moving. So in the case of respiration, things move from a high energy molecule like glucose to a molecule that accepts the electron. The same thing goes for fermentation. It's just all about electrons moving from a high energy molecule like glucose to another molecule that accepts that electron. The difference, though, is the molecule that's used as the electron acceptor. So whenever you're talking about respiration, you're talking about a molecule that uses something inorganic as its final electron acceptor. And so an example of an inorganic molecule that can be used as an electron acceptor during respiration is oxygen. Fermentation is a different process because the things that fermenting cells use as an electron acceptor are organic things. And so by organic molecule, what we mean is a molecule that has both carbons and hydrogens in it. And so an example of an organic molecule that can be used as an electron acceptor during fermentation is this molecule that's called pyruvate. And so that's the thing that defines the difference between respiration and fermentation, is that the molecule that's used as the electron acceptor is different. It turns out that inorganic things like oxygen have stronger electronegativities as compared to things that are used by fermenting cells as their electron acceptor, like pyruvate. And so because things like oxygen have a higher electronegativity, relatively speaking, it means that it's tugging on that electron with more force as compared to the electron that's being moved during fermentation. And so because this electron is basically moving with more force than this electron, it means that there's more kinetic energy that's being generated over here as compared to over here. And so because respiration generates electrons that have more kinetic energy, that means that the ATP that's produced during respiration, there's a lot more that's being produced during respiration as compared to during fermentation, because again, the electrons that are moving during this process don't generate as much kinetic energy, which means that the amount of ATP that's produced in total isn't as much as compared to during respiration. So what we'll do here in this video is talk about what occurs during respiration. We'll leave fermentation for class, so that's when we'll take a look at it. And so here we go. So we'll start with the big picture of what happens during respiration, and then we'll add in kind of one detail at a time. So whenever you're talking about cellular respiration in eukaryotic cells, again, basically what's happening is that electrons are moving from a high energy molecule like glucose towards an inorganic molecule like oxygen. Whenever that electron moves, that is kinetic energy that can be used to make something else that has a lot of energy like ATP. So again, that's the essence of cellular respiration. It turns out that there's some details though, so there's stuff that that occurs in between here. And so one detail that we'll add in is this. It turns out that the electron that moves between glucose and oxygen, it doesn't go directly from the glucose to the oxygen. Instead, the electron that's delivered goes through a series of molecules that's together known as the electron transport chain. And it turns out that the electron transport chain in eukaryotic cells is found within the inner membrane of the mitochondria. So this makes sense. The electron transport chain is involved in making ATP, and if you remember from a few weeks back, the mitochondrion is the organelle that's responsible for making a lot of ATP. So it makes sense that the electron chain is found within the mitochondrion. Again, the electron that's transferred from glucose ends up running through this electron transport chain before the electron gets to the oxygen. And it turns out that it's at the electron transport chain that the energy of the moving electron is then used to make ATP energy. Here's another little detail that we'll add in. So it turns out that in order to get the electron out of that glucose molecule, you basically have to break that glucose molecule apart. And so one major way that cells have in order to break apart that glucose is through something called glycolysis. And so glyco, you might remember that prefix means sugar. Lysis means to break. So literally this means sugar breaking, and that's exactly what happens during glycolysis. You have a molecule of glucose that's basically broken in half. It turns out that during that process of breaking apart a glucose molecule, that generates a little bit of ATPs on its own. But more important, and the reason why it's more important is because this is what generates a lot more ATP, the breaking apart of that glucose releases high energy electrons, and it's these high energy electrons that end up being delivered to where the electron transport chain in the oxygen is. It turns out that there's actually a molecule that's responsible for making sure that that electron gets to where it needs to be. So electrons, because of that negative charge, could potentially cling onto anything that has a slight, even the slightest positive charge to it. So if you're interested in making ATP, you really want to make sure that the electron gets to where the electron transport chain is. And so the name of the molecule whose job it is to deliver that electron to the electron transport chain, it's called nicotinamide adenine dinucleotide, or NAD+. And so basically NAD+, it's an electron limousine that takes the electron to where it needs to be. It turns out that NAD+ not only takes on electrons but also protons. So you might remember again that an electron and a proton equals a hydrogen atom. So what we're saying here is that NAD+, it's the empty version of the electron limousine. After it takes on electrons and a proton, it becomes NADH. And NADH is the loaded, or the full version of the electron limousine. So it's this full electron limousine then that ends up delivering its electron cargo, which is in that hydrogen atom, to where the electron transport chain is. And whenever that the electron that's in that hydrogen moves through the electron chain towards the oxygen, again, it's that kinetic energy of the moving electron that's used to make ATP. You see these leftovers of glycolysis here. So glycolysis is the breaking in half of a glucose molecule. So here are the two halves. Those are called pyruvates. And it turns out that even more electrons can be squeezed out of these pyruvates. And so where that occurs is in something called the citric acid cycle. And the citric acid cycle occurs in the it occurs inside the mitochondrion. So the electron chain happens in the inner membrane of the mitochondrion. Glycolysis happens outside of the mitochondrion in the cell's cytoplasm. The citric acid cycle happens kind of in the cytoplasm of the mitochondrion. So again, these pyruvates can have even more electrons squeezed out of them during the citric acid cycle. It turns out, though, that these pyruvates can't enter directly into the citric acid cycle. So first they have to be converted to a slightly different molecule that can then enter the citric acid cycle. And so here's what we mean by that. So again, before these pyruvates can enter the citric acid cycle, actually what occurs is that one of the carbons ends up being removed from the pyruvate in the form of a CO2 molecule. So you know the CO2 that you end up breathing out as a result of cellular respiration, that's one of the places where the CO2 comes from. That carbon that was removed from the pyruvate is replaced by something called coenzyme A. And so whenever you have this molecule now, two carbons and a coenzyme A, these molecules are called acetyl coenzyme A's. And it's the acetyl-CoAs that can then enter the citric acid cycle. It turns out that during the citric acid cycle, a little bit of ATP molecules are made, but maybe more importantly, high energy electrons are squeezed out of these acetyl-CoA molecules. And again, these are electrons that can be delivered to the electron transport chain where a whole mess of ATPs can be made. Again, there's a molecule that serves as an electron limousine that makes sure that these electrons get to their destination. Right, so we already mentioned that NAD+ is an electron limousine, so it takes on an electron and actually two electrons and a proton to become NADH. NADH then delivers its electron cargo to the electron transport chain. Once the electron in that hydrogen atom moves through the chain, that generates the kinetic energy that's required to make ATP. It turns out that there's also another electron limousine that can work together with the citric acid cycle in order to deliver electrons to the electron transport chain. And so the slightly different version of an electron limousine that can work together with the citric acid cycle is something called FAD. So FAD can also take electrons and proton actually to become FADH2, and it can also then deliver electrons to the electron transport chain in the form of a hydrogen atom. And again, when the electron in that hydrogen atom moves through the electron chain, that generates the kinetic energy that's required in order to make ATP. Right? So when you're talking about respiration in a nutshell, this is it. So most of the ATPs are made when electrons move through the oxygen. The electrons ultimately come from something like glucose, but the electrons that are in that glucose have to be released. Those released electrons then are delivered to the electron transport chain by either something called the NAD or FAD, if you're talking about the citric acid cycle. So what we'll do next is take a little bit closer look at glycolysis, a little bit closer look at the citric acid cycle, and actually in class we'll take a little bit closer look at the electron transport chain. So first we'll start with glycolysis and take a look at more of the details of what occurs during glycolysis. But before we do that, I think it's worth mentioning one thing. So here we're taking a look at basically how respiration occurs in eukaryotic cells. You know that prokaryotic cells, they can also make ATP, and so there are prokaryotic cells that can also do respiration. There is a difference, though, in where glycolysis, citric acid, and ETC is found in eukaryotic cells versus in prokaryotic cells. And so here's that difference. So by definition, eukaryotic cells have organelles, so things like mitochondria. And we've already seen that in a eukaryotic cell, glycolysis occurs out here in the cytoplasm. The citric acid cycle occurs inside the mitochondrion, in the matrix of the mitochondrion. and the electron transport chain occurs in the inner membrane of the mitochondrion. So these things also occur in prokaryotic cells, but prokaryotic cells don't have mitochondria, so there's less separation of these three things that occurs in prokaryotic cells. So in a prokaryotic cell, glycolysis, like in eukaryotic cells, happens in the cytoplasm. The citric acid cycle, because there's no mitochondria in prokaryotic cells, the citric acid cycle just occurs kind of out in the open in the cytoplasm of the cell also. The electron transport chain occurs in the cell membrane itself of the prokaryotic cell. So again, the point here is that prokaryotes also do these things just like eukaryotic cells do, but because they lack mitochondria, those three processes just happen in different spots in a prokaryotic cell. So again, what we'll do next is take a closer look at what happens during glycolysis, the citric acid cycle. We'll save the electron transport chain for class. So first we'll take a look at glycolysis. And so again, basically what occurs during glycolysis is that you have a 6-carbon molecule, glucose, that's broken in half, and each one of the two halves are called pyruvates. We'll color code this glucose molecule so that the first 3 carbons are one color and the last 3 carbons are another color, and we'll see why it's good to color code them here in a minute. So here's how we get from here to here during glycolysis. So the first thing that happens to that glucose molecule is that actually phosphate groups are added to either end of that 6-carbon molecule. And so you might be wondering, where does that phosphate group come from? It actually comes from an ATP molecule. So during these first few parts of glycolysis, an ATP molecule is actually required. And again, why it's required is because one of its phosphate groups is added on to that glucose molecule. So that here is now that phosphate group. After that ATP gives up one of its phosphates to the glucose, that ATP now has one less phosphate group. So now it's an adenosine diphosphate, an ADP, instead of an ATP. That's where this phosphate group comes from as well. So this phosphate group also comes from an ATP that comes in during the first few parts of glycolysis to donate a phosphate group and then leave with one less phosphate group. So it leaves as an ADP molecule. So you're seeing here that during glycolysis, actually ATP is required during the first kind of initial stages. Here's what happens next in order to eventually release, in order to make high energy molecules. So this molecule is actually then broken in half. It turns out that this portion, so this half of the glucose molecule, is the only half that can continue on with glycolysis in order to produce pyruvate. So here I know they look the same. We've kind of simplified this so that we're only looking at the carbon skeleton. But if you were to take a look at the complete structure of glucose, you'll see that this half has a little bit different structure as compared to this half. And so that's, again, so these are different. It turns out that this is the only half that can continue on to form pyruvate. But kind of the good news is that this version of the 3-carbon molecule can easily be converted into this version. Right? So this can be converted into, or sorry, this can be converted to this. So because this can be converted to this, essentially what you have after the first stage of glycolysis are 2 molecules that could each then enter the second phase of glycolysis in order to make pyruvates. And so here's what happens during that second phase of glycolysis. The first thing that happens is that actually another phosphate group is added onto these molecules. So now you have 3-carbon molecules that have 2 phosphates on them. Where that phosphate group comes from, it comes from an inorganic phosphate. So an inorganic phosphate comes in, and that's where this second phosphate group comes from. By inorganic phosphate, all we mean is it's a phosphate that's attached to neither hydrogen nor carbon. Right? So this phosphate group up here, it's considered to be an organic phosphate because it's attached to this sugar group that has carbons and hydrogens in it. This phosphate down here is considered an inorganic phosphate because it's, again, attached to neither carbons nor hydrogens. So it's kind of a free-floating phosphate that's used to be tacked on to this 3-carbon molecule. So the same thing here. This 3-carbon molecule acquires another phosphate as a result of an inorganic phosphate joining their fray. And so then what happens to these 3-carbon molecules next is that the phosphates basically are stripped away from them in order to produce pyruvate. The phosphate groups that are taken off are added on to an ADP molecule to produce ATP. Right, so this can be added on to an ADP to make ATP. This can be added on to an ADP to make ATP. So in this little, during this little arrow step here, that actually produces 2 ATP molecules. Same thing here, each one of these phosphates can be used to produce an ATP molecule. And so you see at the end, if you were to tally up the net total of ATPs that are being made during glycolysis, you see that 1, 2, 3, 4 are produced. 1, 2 were actually required at the beginning, so 4 minus 2 means that there is a net total of 2 ATPs that are produced during glycolysis. More importantly, remember that glycolysis also produces, it releases electrons that can be delivered to the electron transport chain. And so the specific step where that electron is released is this step right here. So it turns out that an electron is released during this step. That released electron is picked up by the electron limousine, NAD+. So NAD+ comes in during this particular reaction, picks up the electron to become NADH, and the same thing happens here. This is basically the same step. There's an electron that's released. It's picked up by the electron limousine, NAD+, to become the loaded electron limousine, NADH. And so again, glycolysis, it's all about breaking a glucose sugar in half to form 2 pyruvates. During the process, these really important molecules are produced. NADH goes to the electron chain to make a lot more ATP, and there's also a small amount of ATP that's made directly during glycolysis. So that's glycolysis in a nutshell. What we'll do next is show you a little bit more detailed figure of what occurs during glycolysis. So again, here we've kind of simplified things so that all we showed was the carbon skeleton of each one of the molecules that's involved during glycolysis. So what we'll do next is show you a figure showing the full structures of each one of the molecules involved during glycolysis. And so here it is. Again, glycolysis, you can think of it as being divided into 2 phases. And so here's the first phase, where the glucose is split in half to form 2 molecules that each have 3 carbons on them. Each one of these 3 carbon molecules have a phosphate on it. So here's a phosphate, here's a phosphate. This is the only, of these 2, this is the only 3 carbon molecule that can then enter the second phase of glycolysis. But again, it turns out that this can be easily converted into this. So basically what you have are 2 molecules then that could enter the second phase of glycolysis, and so that's why you see a 2 in front of everything that happens during the second phase, is to let you know that basically the second phase happens twice for every glucose molecule, right? Because again, both of these can enter the second phase. And again, what occurs during the second phase is that eventually the phosphate groups that are on each one of those molecules are used to make ATP molecules, and then at the end, basically what you have as one of the leftovers is pyruvate. So this matches up with that, that kind of basic picture that we showed you before. What we'll point out here, now that you can actually see the structures, is this. So it turns out that during this first phase of glycolysis, you see that the glucose molecule actually ends up getting rearranged a little bit. So here you have a 6-carbon molecule, glucose, and here you also have a 6-carbon molecule. The difference between this and this is that here 5 of the carbons are found inside of this ring structure, and then the 6th one is outside. Here 4 of the carbons are found within the ring structure, and then you have 2, this one and then this one outside. And so it turns out that this is a molecule of fructose. This is a molecule of glucose. So during this first phase of glycolysis, glucose actually gets converted into fructose before it kind of gets 2 phosphate groups then on it eventually. The thing we'll mention about the second phase of glycolysis is that there's some rearrangements that occur to that 3-carbon molecule. Right? So one of the first rearrangements, or one of the arrangements that you see, occurs at this step where the phosphate ends up switching places. Right? So in this molecule, the phosphate is on this end carbon, whereas in this molecule, the phosphate kind of jumps up to that second carbon. So that's kind of one rearrangement that occurs, and then another rearrangement happens in this step where you end up seeing a double bond form here, where it was not seen here. So basically what these rearrangements do is that they kind of destabilize that phosphate to make it easier for it to come off in order to be added to an ADP in order to make ATP. So again, glycolysis, it makes some ATPs. It makes some really important NADHs that go to the electron transport chain. If you take a look at the full structures of all of the molecules involved during glycolysis, you'll see that there's some rearrangements that occur in order for that to happen.